Hello, everybody, and thanks for joining us. I'd also like to take a minute to introduce the session. This is an educational session. So the point and the goal of this session is really to provide a broad introduction to a new field, which some of you are new to, which is genome editing. And we basically subdivided the whole field into 3 different modalities. The first one is DNA nucleases, which I will cover. Then we talk about base editing, which is a very hot field right now. And finally, we talk about piggyback transposon-mediated cut and paste mechanisms. So this is my talk, DNA nucleases in gene editing. My disclosure is a full-time employee of Grace Science. So here's an outline of my talk. So I will basically cover quite a lot of ground and basically cover, first of all, an overview of nuclease-based genome editing, talk about tools, technologies, platforms, and approaches, but also some of the applications and preclinical and clinical development. Then we'll get a little bit deeper into the role of DNA repair mechanisms in genome editing, which I think is a little bit underserved usually. And then I will talk a little bit about nuclease-free genome editing and what the potential is of that technology. And finally, I'll talk a bit about ethical concerns of genome editing, which is sort of a recurring theme every year at this meeting. So to begin with, first, we'd like to distinguish somatic genome editing from germline editing, which we'll cover at the end of this session. So somatic genome editing can either be done to gene replacement or gene disruption strategies. And you can either do this ex vivo or in vivo. And on the right, you can see all the different tissues that have basically to date been targeted either in preclinical or even in clinical studies, like the CNS, the eye, the liver, the muscle, but also hematopoietic stem cells. The delivery to non-germline tissues usually gets mediated by viruses, mostly AAV, which different serotypes confer different delivery specificities. In the past, also adenovirus has been used, and this is usually to deliver DNA. For mRNA delivery, we also have lipid nanoparticles, which come of age, and there's sort of extra sessions on this. We have ribonuclear proteins, but we also have experimental technologies like exosomes, combinations of nanoparticles with AAV, and they obviously can be used to deliver mRNA or DNA. The age of intervention in somatic genome editing depends a little bit on the disease onset, depends on the severity of the disease, but of course also regulatory guidance, and the FDA is involved in this. And this is very important for highly proliferative tissue. And here basically I put together a comparison between AAV-mediated gene therapy and genome editing, where I highlighted the difference and the advantages of genome editing with respect, for example, to the liver. And this is important because the liver grows very quickly in the beginning of pediatric development, in particular the first 2 years, exponentially, and then more linear afterwards. And this means if you use gene therapy, conventional gene therapy, basically you have only a vector dilution after a while, which kind of cuts your efficacy. In contrast to that, and this of course is because the gene therapy is basically based on episomal AAV transgenes sticking around and being expressed. In contrast to that, integrated therapeutic transgene for genome editing provides potentially a lifelong benefit. This of course depends also if you hit stem cells or progenitor cells. The disadvantage is that you have at least one AAV vector at a minimum, but usually you also need to provide the nuclease either by mRNA or an additional AAV vector. Another feature of AAV genome editing over gene therapy is it can provide a knockout strategy as opposed to just a knockdown strategy by siRNA or transcription factors. And in both cases, we have multiple clinical studies ongoing, which are targeting, for the case of genome editing, either the liver, the eye, or also stem cells. So these are sort of the 4 major platforms you will encounter at this meeting. Meganucleases, which is sort of the oldest one with ICE1, popularized by Maria Jason. Then we have zinc finger proteins, which are sort of naturally occurring transcription factors, which you can engineer in these long arrays to bind specifically DNA sequences. And you can then combine them in this sort of dimer here with a FOC1 nuclease in the beginning, in the middle, that sort of introduces a double-strand break because they're guided by the zinc finger proteins. Tailends are very similar. They're proteins specifically binding DNA sequences. And again, people have basically engineered these tailends in combination with FOC1 nucleases to target specific sites in the genome. And then, of course, this CRISPR-Cas9, which basically uses the binding of an mRNA, a guide RNA, to perform a homology search in the genome, and then basically introduce a double-strand break by its intrinsic RAFC nuclease function. And of course, it's not only Cas9, there's a whole sprawling universe of CRISPR proteins now, which can target either DNA or RNA. And they basically have naturally evolved to that point. Of course, now they're being engineered to basically aid nuclease-mediated genome editing. What's very important for this is to look really at how the DNA cleavage is different from all these different systems. For example, Cas9 tends to make mostly blunt ends, which lend themselves more towards HDR recombination, while Cas12 has more 5' overhangs. And then the Fock nuclease, which usually gets combined either with CFNs or tail ends, but can also be attached to a catalytic dead Cas9, usually causes 5' overhangs for nucleotides. And this is important for some of the stuff I'm going to talk later about DNA repair. If you look at the clinic, this is just a quick search on clinicaltrials.com and what basically is recruiting or in phase 1 or 2 right now. There's of course naturally the first trials are from CFNs since it's the oldest technology, and they basically performed the first time ex vivo genome editing using CCR5. This renders the T cells resistant to HIV infection, and this is a study that's now in phase 2. Then we have the albumin approach, which was reported just this morning by Paul Hametz on MPS1. And this is basically a transgene integration mediated by CFNs at the albumin locus for therapeutics in MPS1, MPS2, and hemophilia B. And this is the first evidence for in vivo genome editing. Sangam also developed beta thalassemia. This is a mRNA play for reactivation of fetal gamma globin. And this basically is in phase 1-2 right now. Tailends, mostly like a CAR-T game right now for selectors to basically deliver the CAR-T receptor. And this is, there's also some more exotic applications. I found something called a tailing CRISPR gel, which is presumably topical to sort of excise the HPV genome from neoplasias. So this is more an exotic one in there. And then the CRISPR-Cas9 now has also the first trials on the way. Recruiting right now is a CRISPR therapeutics trial for beta thalassemia, very similar play like the Sangamo program to reactivate fetal globin. Then we have in using AAV5, sponsored by Editas Medicine, and this is sort of the first in vivo gene editing mediated by Cas9. And this is basically excision of a cryptic splice site. And then finally we have just the first of many different CAR-T plays using CRISPR-Cas9 that will come in the future. I mean, this really demonstrates, like, once these technologies now enter the clinic, much more important is not the safety and how we basically assess off-targets of all these different platforms. This is sort of a high-level overview of what can be done and has been done. There's first in silico and CLEX are included there as well. And these are basically biased methods that use the sequence information you have from the guide RNA to look for similar sites in the genome. And there's a whole bunch of those you can use. The problem is they're biased towards the sequence, and they also cannot really predict most of the off-targets that you can find with other methods. And I think that's why these methods should be really limited to research, not to preclinical or clinical use. Then are the in vitro systems. They basically use sort of naked DNA and protein and combine them, for example, circle-seq. And there, have a pretty good sensitivity. You have pretty high modification levels. And the problem is there's no histones presence. That means you're not assessing the off-target activity in the proper chromatin context. So this is fixed in like the in-cellular methods, for example, GuideSeq. And in this case, you basically deliver all these vectors either by mRNA, DNA, or AAV into the cell, which of course gives you lower efficiency, but at least you have it in a chromatin context. So it's important that you don't use cancer cells necessarily, but you also do this in primary cells, since the chromatin context can be widely different, and cancer cells often have a different karyotype. In addition to this, there's other methods to sort of overcome that limitation of these methods, that they can only detect small insertions and deletions. For example, whole gene sequencing, but of course you have to do this many times to sort of get a very quantitative readout. There's PCR-based methods to detect translocations. If you know off-targets, potential off-targets, you can design your PCR to detect those. There's transposon-based approaches, which allow you tagmentation and basically much larger reads that allows you to detect inversions, insertions of AAV, and translocations. And Charlie Gersbach has just published a really cool paper on this for Duchenne. And there's single molecule sequencing with PacBio, which again gives you long sequence reads to detect large deletions as well. The bottom line is, there's lots of technologies we can use, and we have to use them before we get in the clinic. And of course, the detection limit of these is nowhere near yet the background mutation rates. So we have to work hard to sort of make sure all these technologies are safe. So the role of DNA repair is important because, as I mentioned, the mutation rate in the genome, the natural one, is about 64 mutations in diploid genome per cell division. This gives you a bit of background what we basically have to approach in terms of sensitivity. This mutation rate is sort of a composite of the background radiation that you all encounter every day, which leads to about half of a double-strand break per cell per year. Some of you come from Europe, so you were basically in the plane for 8 hours. That means you had more cosmic irradiation, which accounts for about, one break in 1,000 cells. But it's not really that much if you think about radiotherapy, where you basically have about 50 double-strand breaks per cell if you get a typical dose of 1 to 2 gray. And just to put this in context, a lethal dose is about 3 to 8 gray, depending on the mortality rate you're looking for. So apart from these sort of outside sources, we also have sort of natural organic sources through cell divisions and so on. We have up to 20,000 single-strand breaks per day per cell. We have all these different chemical modifications, abasic sites, alkylations, oxidations, and deaminations, and some of them lead down the road to about 10 to 50 double-strand breaks per cell. And bottom line is our cells really have evolved to deal with all kinds of damage, and including the damage you're going to throw at them with genome editing. So what I try to do here, basically try to sort of put them in different brackets, which repair pathways are really dealing with different genome editing in cells. For example, the double-strand breaks mediated by nucleases usually look like ionizing radiation or replication stress. And they're usually dealt with non-homologous end joining or HDR-mediated repair. Nuclease-free, I will talk about this a bit later, it's a little bit more complicated, but it's mostly looks like transcription or replication stress, and that's usually dealt with by NER or HDR. Base editing looks a little bit like UV damage, but also the activity of AID in the immune system, which is causing somatic hypermutation and class switch recombination. That means the downstream pathways use the EBR, MMR, and also end joining. And finally, transposons and recombinases, obviously resemble mobile genetic elements, but also the activity of something like RAC2, RAC1 and RAC2, which are sort of co-opted transposases for the immune system. Here the repair events are a little bit less characterized, but maybe Lauren has something to add to that. She says no. So for now I want to just take a little deeper dive into the nuclease-mediated repair. And for this, this is basically what you see probably at this talk, this meeting very often. So you see basically on the right side, depending on the phase the cell is in, you can integrate a transgene donor with homology arms by homologous recombination seamlessly into the genome. In contrast, non-homologous end joining can grab that transgene and integrate it, but the homology arms are basically integrated as well, so you have a duplication there. In the absence of a donor, you can actually have like a sticky end cut like here, like we said earlier, for example, by FOG1. This can lead either to wild-type allele or also to insertions and deletions. This gets a little bit more complicated if you look at the pathways that really work on this. And I think end joining always has a bit of a bad rap at this meeting. And I want to basically mention that I think end joining is very much error-free and usually is able to sort of bring the sticky ends together and repair the site error-free. And this actually has been backed up by multiple studies. One of them are from the Wolf lab I'm going to show here. They basically engineered 2 Cas9s in proximity to each other, and he was able to show that the junctions of these double cuts are actually in 99% of the cases precise. It means no insertions and deletions. And he also showed that you can actually have the site up to 200 base pairs apart from each other. So this is just one example where we show that classic end joining is actually not error prone. Apart from the classic end joining, you get usually other repair proteins involved. Some of them are error prone, alternative end joining, but there's also microhomology end joining, which sort of uses 5 nucleotides to sort of eliminate the sequence in between. For homology-dependent repair, you usually provide either a single-stranded oligo, where you can basically integrate it by a single-strand template repair, or the double-strand donor, which is sort of your classic gene replacement. And this usually happens through synthesis-dependent strand annealing. The holiday junctions probably don't play a big role. And I show this because it's interesting, because the proteins involved in all these different pathways actually have been targeted by cancer therapy companies for synthetic lethality. For example, BRCA2, which is here on the right in the middle, is basically a very important protein for HDR repair right here. And if you have basically a BRCA2 mutation, you can basically inhibit one of the enzymes over here on the left, PARP1, and therefore these cancer cells are more susceptible to chemotherapy. And this is really something in the clinic right now for breast cancer and ovarian cancer. And there's many more companies that target all the proteins basically highlighted in the colors here for cancer therapy. But we can also use these inhibitors to basically study the integration mechanisms of genome editing, but we can also try maybe one that use them to sort of shift the balance from one side to the other. for example, PARP inhibition and DNA PK inhibition would shift it towards the HDR. So next I want to go over to nuclease-free genome editing and how some of these concepts apply to this as well. The first one I'm going to talk about is a fairly old technology developed in the Glaser lab in Yale. This basically uses like the induction of DNA triplex structures using peptide nucleic acid, sort of to pry open the DNA and basically force your repair mechanisms to sort of introduce a double-strand break. This is basically because the displaced strand is recognized by endogenous repair pathways, and then two cuts on the left and the right by the XP proteins can actually, introduce a double-strand break, which then can lead to integration. So this is one way to think about nuclease-free genome editing. The most extreme case is where you basically don't use even a nuclease or something to introduce a double-strand break. You just take advantage of spontaneous breaks. And this is work from Marquet lab from 2015, where he showed that you can integrate a promoterless factor IX donor delivered by AAV8 into the albumin locus. Very similar approach what Sangamo did, but with nucleases. It's much less efficient. He showed about 0.5% integration, but he was able to sort of express therapeutic levels of factor IX in these mice. However, the group never provided really a mechanism or data in human cells. And some of this was done better in a study from 2018 by Homology Medicine, where they showed they can do integration not only in human cells, but they also did some mechanistic studies and showed the integration depends on BRCA2, which is sort of the protein I mentioned it earlier, important for HDR. They also showed this technology could potentially be translated into the clinic, where they basically show proof of concept data for gene correction at the PAH locus, which is mutated in phenylketonuria. I think my only problem with these technologies is always that nobody really talks about mechanism and how sort of these breaks are regenerated. And I think there's a good model in the repair field that can be used to sort of study this a bit more. And one of the models is that you basically have collisions between transcription forks, replication forks and transcription bubbles, which can lead to sort of the formation of replication fork collapse. But there's also the idea that the mRNA coming out of a polymerase, RNA polymerase, can actually form so-called RNA-DNA hybrids. And these are also, can basically be the substrate for two nucleases, turning them into a double-strand break. This has been shown by work from Colleen Simbridge in Stanford. So to summarize sort of the nuclease-free genome editing part, there's lots of pros. Obviously, there's the promise that there are few off-targets. There's the promise that you only need to deliver one AAV vector, the transgene, for integration, for efficient integration. And of course, this would have advantages for the delivery efficiency and manufacturing. The concept at this point is that you have overall lower efficiency, and some of the approaches actually rely on selection. Also today, there's no large animal data yet. We have some effects could be due to ITR activation. People have shown the ITR has some intrinsic transactivation activity, and of course these effects would not be durable. There's the problem of the unclear double-strand break generation prior to integration, and especially the R-loop idea would really only apply to highly transcribed loci. That means your gene editing will really be applicable only to very few loci in the genome. And finally, what can happen once can happen twice. That means spontaneous break in that area could potentially inactivate your transgene. So the summary is that a lot of work could be done and has to be done sort of to make this really fulfill its full potential. So at the end, I'd like to take a pause and just talk a bit about some of the ethical concerns of genome editing. And this is really something that Hank Greeley, like a bioethicist from Stanford, said a few years ago at ASGCT, that there's always these sort of like cycles of anxiety associated with technological progress. And that happens, periodically. And it started really for gene editing in 1975, and Paul Burke delivered DNA using SV40 into mammalian cells. And there was a moratorium at the time for experiments, and which led to this Acelluma conference, which is often quoted. And eventually it led actually to the creation of the Recombinant DNA Advisory Committee, or short RAC, which was actually pretty much replaced by another committee just 2 weeks ago. It's now called NextRAC. It means basically gene editing came of age and kind of is exiting sort of the RAC oversight. And for a long time, not much happened. And even in 2014, there was discussion at ASGCT in a session with Ruhr Vienisch about germline editing, but everyone's like, this is all being dealt with, this is not an issue. Until then in 2015, Chinese researchers created genetically modified embryos, but did not implant them, which was sort of like, led to an outcry in the West, and basically several papers being published, one of them calling for a moratorium, and the other one basically having a more progressive approach and proposing sort of guidelines how these experiments could be carried out in the future. All this also led to a meeting at the National Academy of Sciences around the corner from here. It was a summit for human genome editing with conjunction with Royal Society and Chinese Academy of Sciences, and they had a quite wobbly statement at the end. I was there and I was a bit surprised how little direction it actually gave. That yes, research is allowed but no implantation, and this was really in contrast to what, for example, the NIH said at the time. They're not funding that research. The FDA does not review any clinical approaches because they're not funded. They're actually defunded by Congress for this. They also said the clinical use of somatic gene editing should not be hindered, and that basically the use of germline editing approaches is only permitted under safety, broad societal consensus, and regulatory oversight. So around about the same time, basically there was also a report putting together which sort of, again, summarized all of this and which is available to everyone. Again, saying no moratorium, enhancement by germline editing should not be pursued, should only be pursued for therapeutic reasons and only under really strict oversight. And there's always the ongoing discussion of what really compelling reasons are. And the fact is there's very few cases actually where pre-implantation diagnostic is not an option. And even sort of the cheerleaders of germline editing are really sort of arguing that way, kind of excluding sort of the fact that embryo editing itself has a very low success rate, and that would also apply here. There's also still unknown issues around safety, mosaicism, and also like the association of germline editing with somatic end editing is kind of potentially problematic as well. Just to finish this off, we basically, at the same time, again, when this sort of was crafted, again, a researcher in China basically finally implanted some of these embryos, and this was actually after modifying the CCR5 locus I mentioned earlier to render sort of these girls resistant to HIV infection. This was in the context that the father was actually HIV positive. Obviously, again, this was done without any oversight, and again, caused quite a stir in the press, but also in the public, and even caused trouble for some Western researchers who actually were aware of this. Up to this point, it's unclear if he actually had produced what he wanted to do, basically rendering these girls HIV resistant, what the level of mosaicism was in these babies. That means which cells actually carry mutations, which don't. And finally, what kind of unintended consequences these treatment had. Since you have off targets, obviously, for all these nucleases, as I mentioned, but even sort of the on-target knockout of CCR5 is not clear how it contributes to normal human development. And I mentioned earlier some Western researchers got in trouble, but of course the problem is there was not really a commission that could have reported this to. And just recently the NIS called basically for the formation of such an international commission overseeing some of these studies worldwide. And of course it's a very lofty goal, but I think it's the right step in the right direction. So to summarize, 2019 is an exciting time to be a genetic engineer. All of you who know Blade Runner, 2019 is the year. Of course, we're 2 years behind because Leon's inception date was 2017, a couple of weeks ago actually. Multiple nuclease-based platforms are currently entering the clinic. The repair pathways, genome editing, could also represent hurdles if they don't do what you want them to do. The nuclease-free genome editing platforms have a lot of work to do and to fulfill their potential. And as I mentioned at the end, the discussion of ethical concerns associated with germline editing has barely begun. But again, it's global consensus is ambitious but a worthwhile goal for this. Acknowledgements, these are just people not redirecting, contributing to the talk, but people who discussions and ideas are in here from Grey Science, Sangamo Therapeutics, and other places as well. I'd like to thank them. And then a little plug at the end. We have an open position at Grey Science for CNS research scientists 1-2. Send me an email. Thank you.